Welcome to AI Explains. Okay, so imagine this. It's December 2019, Wuhan, China. Doctors start seeing all these patients with this really severe pneumonia, and they give them the usual stuff, antibiotics, but nothing works.
And that right there, that was the first huge clue. Because if antibiotics don't work, it's not bacteria. You're hunting something else, probably a brand new virus.
And that's the detective story we're getting into today. We're doing a deep dive into the study by Zhou and his team that was the first to really identify what we now know as SARS-CoV-2.
So let's start with the clues at the crime scene. The patients had these classic symptoms: fever, a really dry cough, and what doctors called dyspnea.
Which is just having trouble breathing, right? Shortness of breath.
Exactly. But the real smoking gun was in the chest scans. They were seeing these things called ground glass opacities.
It sounds super technical, but it's actually pretty descriptive.
It is. Instead of being clear, the lungs on the x-ray looked all hazy, almost like frosted glass. It meant the air sacs were filling up with fluid, the lungs were under attack.
So they know there's an attacker, but they need to get its fingerprints. To do that, they went straight to the virus's instruction manual, its genome.
They sequenced it from patient samples, and that's when the case, it just broke wide open. They found the virus's genetic code was a 96% match to a coronavirus found in bats.
Wow. 96%. In viral terms, that's not a cousin. That's like a sibling.
It's incredibly close. And it was also about an 80% match to the original SARS virus from the early 2000s.
So it's in the same family. They knew what they were dealing with, sort of.
Right. It placed the suspect firmly in that family tree and told them this almost certainly came from bats.
Okay, so they have a suspect. But every good detective story needs to know the weapon. How does this thing actually break into our cells?
Well, this is where the classic lock and key idea comes in. A virus has a key on its surface. In this case, it's the spike protein, and it has to find the one perfect lock on a human cell to get inside.
And because it was so similar to SARS, they had a pretty good guess what that lock was.
They did. They suspected it was the ACE2 receptor.
That's a protein on our lung cells, isn't it? It's supposed to be there for, like, blood pressure.
It is, but the virus hijacks it, uses it as a secret door. And they proved it with this really neat experiment. They took cells in the lab that do not have that ACE2 lock.
Right, I've heard of these. HeLa cells.
The very same. So they tried to infect the normal HeLa cells, and nothing happened. The virus just bounced off. No lock, no entry.
But then they gave the cells the lock.
Exactly. They engineered the cells to produce ACE2, and suddenly the virus could get in, it could open the door and start replicating. That was the proof. ACE2 was the entry point.
So they have the fingerprints from the genome, and now they know how it breaks in. But they still need to place the suspect at the scene of the crime, right? How did they prove this virus was actually making people sick?
They looked at the body's own defense system, they checked the patient's blood, and they found neutralizing antibodies.
The body's soldiers.
The body's soldiers trained for one specific enemy. The fact that the patients were making antibodies specifically for this new virus, that proved this virus was the cause of the battle raging in their lungs.
It's just incredible. This one paper gave us the first mugshot, the first fingerprints. It's what allowed everyone to start building tests to actually find this thing.
It was the bedrock for the entire global response, really.
But the paper ends with a bit of a warning, doesn't it? The authors link the outbreak to wildlife and say bats are carrying a lot more viruses like this one.
They do. Their final thought is, a pretty serious one. They suggest that how we interact with wildlife, especially in markets, created this problem. And if we don't change that, what's to stop another, maybe even more dangerous cousin of this virus from making the same jump?
A really sobering thought to end on. We might have solved this one mystery, but the bigger puzzle is still there. Thanks for listening to this deep dive.